So welcome back from the break. Let's sort out these and put them into order. So if we're talking about animals, the earliest evidence of the animals is the ediacarans. So this would be number, oops, let's fire up the pen. This would be number one. Then the next event would be terrestrial arthropods. So this would be number two. The origin of the mammals would be event number three, because remember the mammals were present throughout the age of the dinosaurs. So when the dinosaurs go extinct would be event number four. So that would be the timeline order of these four events. So now we're going to talk about body plans, because that's the next section. I know that for sure. So when we start thinking in terms of body plans, there are a number of features that we can use to describe animal body plans. And we're going to look at some of these features. So one of the things that we need to do is differentiate between the term grade and the term clade. So remember, a clade is a group of animals that share a common evolutionary origin and includes the common ancestor to that group. A monophyletic group, in other words. That's what we mean by a clade. A grade is simply a group of animals that are grouped together because they share a common feature. So in the past, when biologists and taxonomists have tried to group animals on the basis of body plans, because remember, that was what they were really using. What they were really creating was not a clade, but a grade because of shared body plan features. So we're going to take a look at some of those features. And what I'll do as I go through these is point out why the groupings were created, but also why they no longer serve the purpose of forming keys for creating taxonomic groups. So the first feature is symmetry, and we recognize two basic types of symmetry: radial symmetry and bilateral symmetry. So the radial symmetry was always considered to be the simplest of the two body plan forms, and the phyla that contain radially symmetrical animals that are true radially symmetrical animals have always been placed basal to the rest of the animal kingdom. So these would be the cnidarians, and ctenophorans. Both show radial symmetry, and as you'll see in a few minutes, both also show diploblastic organization during gastrulation. In other words, two layers of germ tissue, ectoderm and endoderm, which remember correlates with radial symmetry. Now, of course, there are other clades or other groupings, phyla, that we know show radial symmetry. Specifically the echinoderms, so the sea stars, the sea urchins, and so forth. But that's a product of metamorphosis from a larval stage to an adult that has radial symmetry. But in that larval stage, they show developmental stages that are very much bilateral, and in fact are the same developmental stages that we pass through as bilaterals. And so echinoderms, quite surprisingly, are considered to be the closest phylum to our own as phylum Chordata. And I know that seems kind of strange to say, but that is the case. Based on genetic sequences, based on developmental stages, embryological development, it is pretty clear that echinoderms are perhaps not a sister clade, but a very closely related clade to chordates, and certainly one of the most closely related clades to chordates at the phylum level. So most other phyla so the cnidarians and the ctenophorans are the two big clades or two big phyla that are considered to be radially symmetrical, true radially symmetrical. Most others are bilaterals. So the radial symmetry being the quote unquote simpler body plan as far as symmetry is concerned still is probably a valid observation, because even with the use of the phyloCode and phylogenetics and cladistical analysis, we still place the cnidarians and the ctenophorans, the radiata, to give them their umbrella name, are still placed basal to the rest of the animal kingdom, with one exception, the sponges. Sponges are considered to be even more basal. And with the bilaterals, then we also recognize that there are three axes. We've talked about these before. So the anterior to posterior, head to tail, dorsal ventral, top to bottom or back to front, and then left and right. And it's in fact the symmetry around the anterior to posterior axis, in other words, the left-right symmetry, that is being referred to when we talk about bilateral symmetry. Then all bilaterals to some degree, and the level to which we see this is dependent upon the type of organism, but we see the development of cephalization. Now, the note says here that cephalization is the development of the head. We can make a more general statement here. It's the what's the word I want to use here? Yeah, I'll say it this way: the clustering, sensory organs the anterior end of the animal accompanies development of the anterior end of the CNS into so you do see a progressive increase in the level of cephalization. This development of the brain, which you see with some invertebrates, but you see more highly developed in the chordates, where you actually start to see the development of a brain structure that you and I would recognize, the three components: forebrain, midbrain, and hindbrain, with the central nervous system extending anterior to posterior away from that, and then the encasing of the developing brain in a formal structure, initially made out of cartilage, but then made out of bony material that eventually evolves into a skull, or a cranium, and then a skull. So this is the process of cephalization, and the development of the bilateral body plan then favors the development of cephalization and the creation of a polarity in the body, and obviously the clustering of sensory organs with the development of cephalization makes logical sense, because the head is the first part of the body to encounter a new part of the environment. The head always leads. And so you want to cluster the sensory organs to the head so that you can sense what's in front of you. So that's symmetry. Then the organization of the tissues during gastrulation, specifically the germ layers, into ectoderm, endoderm, and mesoderm in triploblasts, or ectoderm and endoderm in diploblasts. So this would be ectoderm, endoderm. And of course, the three germ layers are ectoderm, mesoderm. And of course, we've tied this before, and let me just make this evident again. I hope this is self-evident, but just to reinforce this notion, if you are diploblastic, if your gastrula is diploblastic, you show radial symmetry. If your gastrula is triploblastic, then you show bilateral symmetry. Then another feature that has been used as a diagnostic for establishing the phylogeny or the taxonomy of the animal kingdom has been the presence or absence of body cavities. So if you are a triploblast, then you have a body cavity that is independent, or generally independent, of the blastocele. So there are basically three forms of body cavity or body plan on the basis of body cavities. So the simplest is this one, what used to be called a coelomate. That's not right. It's single C. Like this, we'll take that out. So, AKA. And so historically, the acoelomate body plan has been considered to be the most primitive. Remember again, if you've got a digestive tract with a cavity in it, that doesn't count as a body cavity. This is a body cavity that's independent of the digestive tract. So the acoelomate body plan, or as it is now referred to as the compact body plan, was considered to be the most primitive. And so animals with an acoelomate body plan were considered to be more basal than those that had a cavity. So the simplest form of cavity is now referred to as the hemocoel body plan, but it has previously been known as a pseudo coelomate. And what identifies a pseudocoelomate or hemocoel body plan is the fact that the body cavity is lined by mesoderm-derived muscle on one side of the cavity only. So the body cavity does not develop from the mesoderm itself. It develops separately from the mesoderm. And typically we say that it develops in a space between the mesoderm and the endoderm. So you can see that here. And so this is considered to be a more advanced body plan than the acoelomate body plan if you're looking at grade analysis. But it is not the most advanced body plan. The most advanced body plan is a true body cavity, or a coelom. So AKA true cavity. So pseudocoelom means false cavity. So the hallmark of a true cavity or a coelom is the fact that the cavity is surrounded by muscle tissue or lined by muscle tissue derived from mesoderm on both sides of the cavity. And some of that mesoderm tissue develops into what is known as mesenchyme, and mesenchyme allows for organs to be suspended within the body cavity, irrespective of whether the body cavity is fluid-filled or air-filled. You can suspend organ systems in that cavity, and mesoderm-derived tissue called mesenchyme is what does the suspension. And this has been considered historically to be the most advanced body plan. But if you consider what we saw in the Cambrian diagram, the presence of a burrowing bilateral worm, and we put that together with the simple observation that the only burrowing bilateral worms that we have extant today are the annelid worms, and they are segmented. They have true body cavities lined with muscle tissue on both sides. They have a very, very advanced, quote unquote, advanced body plan. And yet that body plan is found in the fossil record back in the Cambrian as part of the Cambrian explosion. That suggests, in fact, that this very highly advanced body plan, this segmented coelomate bilateral body plan, actually is a very early animal body plan. So where does that leave this ranking of acoelomate being more primitive than pseudocoelomate, being more primitive than coelomate? Well, it leaves it in a position of not being supportable. And what we now have come to recognize on the basis of genetic sequencing, on the basis of the fossil record in some cases, and a rethinking of the nature of body plans, is the simple recognition that the presence or absence of body cavities, and if a body cavity is present, the nature of that body cavity, is not a good measure of evolutionary history. In fact, what we now recognize, or what we now suspect, is that the segmented coelomate bilateral body plan is the original bilateral body plan, or at least a very early bilateral body plan. And that the pseudocoelomate and acoelomate body plan is a derived characteristic of the animals that possess them as a consequence of their adaptation to the habitat and lifestyle that they happen to occupy. And that is because, remember, that if a feature costs more in terms of the investment of resources and energy than the benefits you get from it, then in an evolutionary sense, you lose it. Not because you can't use it, but because you have to invest too many resources in maintaining the presence of it. So it's not Lamarckian. This is not a use it or lose it. This is, if it's too expensive to keep it, you let go of it. And so for animals that have a pseudocoelomate body plan or an acoelomate body plan, investing in a true body cavity, a coelomate body plan, is just too much of too great an investment of resources and energy, and so they don't maintain those body plans anymore. So body plans are no longer considered to be diagnostic in terms of evolutionary history and evolutionary origins, because loss of a body cavity or absence of a body cavity is not necessarily, evidence of a primitive body plan. It's just an evidence of adaptation to a lifestyle and a habitat. Then the final body plan form that we need to talk about or the body plan form that we need to talk about, is the nature of the development of the body plan. What used to be known as protostome and deuterostome development that gave rise to two clades. So the protostome development gave rise to the clade protostomia, and the deuterostome development gave rise to the deuterostomia. But protostomia is no longer valid. We don't recognize it anymore. We still recognize the deuterostomia, and mammals belong to the deuterostomia as part of that clade, but we simply don't recognize the protostomia anymore. However, the protostome and deuterostome developmental paths are still recognized as being valid. So let's take a look at the various features that relate to that. So the first is the nature of cleavage. In protostomes, the cleavage is said to be spiral. That means that when the third round of cleavage occurs, the cleavage plane, remember, at the third round of cleavage is always horizontal. And because of the nature of the cleavage, the plane causes the upper layer of cells to rotate slightly during that cleavage with respect to the lower layer of cells. And so the cleavage planes become offset. And what you find is that as more and more of the horizontal cleavage planes occur, you get a further and further rotation of the upper layers of cells with respect to the lower layers of cells. Each horizontal cleavage results in a rotation of the cells above that cleavage plane with respect to the cells below that cleavage plane. And so you get a development of a blastula, because it is basically the early stages of the development of the blastula, that has layers of cells that are offset from each other. The notion of determinate cell division means that for many of the clades that belong to or that follow the protostome developmental pathway, as the blastomeres start to form, starting with the first cleavage, the resulting blastomeres lose the ability to become any part of the developing embryo. In other words, they lose their potency. So determinant means loss of totipotency in the resulting blastomeres. So if you recall, when we talked about this in the previous presentation, I was trying to think of the stage in between totipotency and determination, where the fate of the cells is fixed. And I finally remembered it. It's pluripotent. So we go from totipotent to pluripotent to determined. So the loss of potency in protostomes occurs as soon as cleavage begins. Now in deuterostomes, when the third plane of cleavage goes in, that first horizontal plane, the nature of the cleavage is such that the cells do not rotate. You get a complete cleavage across the layer, and because there is no offsetting of the cells, the vertical cleavage planes remain aligned with each other. And this is also true with subsequent horizontal cleavage planes. There is no offsetting of the layers of cells above the cleavage plane with respect to the layers of cells below the cleavage plane. So the vertical cleavage planes remain in alignment. And when we say indeterminate, what we mean here is the resulting blastomeres remain totipotent. Until gastrulation. In a general sense. Now, we do know, for instance, that in human reproduction, as cleavage takes place and you get the formation of the trophoblast, remember the cells in the trophoblast lose their potency. They're no longer totipotent. In fact, they're no longer involved in the formation of the embryo at all. It's the cells in the inner cell mass that retain the totipotency and will contribute, or some of them at least, will contribute to the formation of the embryo. So there's no one-size-fits-all definition of determinate versus indeterminate, but there is this general sense that in determinate growth, the potency is lost essentially immediately the blastomeres start to form during cleavage, whereas in indeterminate growth, the potency of the blastomeres is retained for a longer period of time, although we know that at some point, usually around gastrulation when the germ layers form, that potency is now compromised because the germ layers are not going to become all types of cells. They're starting down the pathway to determination, so they would be at that point pluripotent. Then in the situations where a body cavity does exist, a coelom, a true body cavity, in protostomes, the body cavity formed as a result of a splitting of the mesoderm, and this is referred to as a schizocoelous body cavity. It's a consequence. Schizo means splitting. So you have a splitting of the mesoderm to give rise to the cavity that will become the body cavity. In deuterostomes, the body cavity is a result of a pocketing of the mesoderm associated with the archenteron. And this is referred to as an enterocoelous body cavity. Because it's derived from a pocketing of the archenteron, the primitive gut, enteron referring to gut, arc, primitive. So an enterocoelous body cavity is a body cavity that is derived from the gut cavity via this pocketing in the mesoderm that's associated with the gut cavity of the developing deuterostome embryo. So the origin of the body cavity is different in protostomes versus deuterostomes. However, the big feature or the defining feature, the one that is the bespoke feature, the one that gives it its name, is the fate of the blastopore. So in a protostome, proto meaning first, coming from the Greek word protos, and stome or stoma meaning mouth. Oops, That's not what I wanted. Let's that and do this again. So let's just take out everything and start again. So protos means first, and stoma I think it's stoma, it could be stoma or stomate, means mouth. So the blastopore the blastopore as the first opening of the archenteron becomes the mouth. So deuterome means second. So in this case, in a deuterostome, the blastopore doesn't become the mouth, it becomes the anus. The second opening becomes the mouth. In a protostome, the first opening becomes the mouth, the second opening becomes the anus. So this is the bespoke feature that gives the names to the two developmental paths, the protostome development versus the deuterostome development. So let's take another break. This is our last break before the end of the presentation. I said this was a short one, and it is a very short presentation. So let's take a look at this concept check question, then we'll take a break, and I'll meet you on the other side with the answers to these questions. So compare the early development of a snail, protostome, with that of a human, a deuterostome. And how do animals that lack a body cavity, the so-called acoelomates, or compact body plane, exchange materials? See you on the other side of the break with the answers.